Hello everyone, today we have the continuation of our lab and it is about the results that we get after we incubated our cells. And today we are going to isolate GFP protein from our cells. After the overnight incubation, we remove the tubes from the shaker. Now we need to observe the GFP expression under the UV light. You could see the transformed bacteria tilted to the right. While the bacteria that were not transformed tilted to the left. Close the lid, wear the protective equipment, the face shield, and turn on the lamp. In darkness, you could see that non-transformed bacteria are not glowing, while the right tube is glowing. Today we are going to proceed with the purification of GFP, and we are going to use the hydrophobic interaction column chromatography, and for this activity we would need one column which contains the hydrophobic material inside. We will need the bacterial cells grown in the incubator and also the set of solutions. First one is tris EDTA buffer or TE buffer. It contains 1 millimolar EDTA and 10 millimolar tris with a stable pH of 8. We would need solution that contain ammonium sulfate of different concentrations. Those include equilibration buffer, which is 2 molar ammonium sulfate in TE buffer, then the binding buffer, which is 4 molar ammonium sulfate in tris EDTA, and finally the wash buffer, which is 1.3 molar ammonium sulfate in TE buffer. We would also need the centrifuge tubes, pipettes and pipette tips. Finally, we would need the important part of today's experiment, the centrifuge. We are going to use the microcentrifuge available at our lab. We start with the transferring of 2 ml of overnight grown culture into the centrifugation tube. Make sure that you mix the solution well before you transfer it to the tube. Now we are going to place the microcentrifuge tubes into the centrifuge and also balance them to make sure that the distortion does not apply to the rotor. To balance, just put the same volume liquid on the other side, and we're going to use the RCF units here, so it's 12.3 G. And also we set the timer for 5 minutes. Following centrifugation, observe the sample. You could see that on the bottom of the tube there is a pellet of the cells. These are precipitated cells. Above it you could see the liquidous part, which is the supernatant. We don't need it. The next part is to remove the supernatant and observe the pellet under the UV light. As you can see, the pellet is glowing. Now we need to resuspend the bacteria, and for that we are going to use 250 microliters of TE buffer. Pipette it up and down slowly to resuspend all the cells in the pellet. After resuspension of the cells, we are going to add a drop of lysozyme. Lysozyme is the enzyme that is going to attack the bacterial cell wall. It will help us in the destruction of our cells and releasing GFP into the solution. Using Pasteur's pipette, transfer lysozyme into the tube. Now mix it gently by flicking and place the centrifuge into the freezer. Remove your frozen sample from the freezer and warm it up with your hands. The cycles of freezing and thawing will help in the breakdown of the plasma membrane. Now we need to centrifuge bacteria for 10 minutes at highest speed, which is 12.3 thousand G. Of course, you need to balance it on the other side of the rotor. While this centrifugation happens, prepare your column. For this, open the top cap and then open the bottom. We are going to use the yellow cap to close the bottom well of the column. Now add 2 ml of equilibration buffer into the column. This is needed to push the hydrophobic beads to the sides of the column. It will prepare the binding of GFP to these hydrophobic beads. Make sure that you do not disturb the column itself with a stream of buffer. Now place the column onto the collection tube and open the bottom part. We are going to let 1 ml of solution drip down. In this case, we will have 1 ml left in the column, and you are going to make sure that it does not stick to the collection tube, otherwise the column will not function and the flow of the solution will not happen with the help of gravity. Now we need to wait until 1 ml is left. After the centrifugation is done, you are going to take your samples and observe them under the UV light. Remove supernatant into a new centrifuge tube because it will contain our GFP protein. The cell debris will stay as a pallet on the bottom of this centrifuge tube. Now add 250 microliters of binding buffer into the supernatant. The binding buffer contains high salt concentration solution of 4 molar ammonium sulfate. Now our samples are ready to be loaded onto the column. Take 3 collection tubes and label them 1, 2, and 3. Place the prepared column onto the collection tube number 1 and open the yellow lid at the bottom. Now we let equilibration solution to drip until it reaches beads. Load 250 microliters of supernatant onto the column and let entire volume pass through the column. Which means waiting until they reach the beads. To do so, open the yellow lid and let it drip into the collection tube number 1. Now transfer the column to the collection tube number 2. We are going to add 250 microliters of washing buffer. The wash buffer should wash out all the unbound proteins that are not as hydrophobic as our GFP, which should bound to hydrophobic beads within the column. Now we are going to open the bottom lid and let the solution go through the column. When there is no solution left above the packing material, that would mean that the 250 microliters pass through the beads. It should take several minutes. Now transfer the column into the collection tube number 3. Now we add 750 microliters of elution buffer, which is the same as TE buffer. Since it does not contain the salts, the GFP will be released from the hydrophobic beads. This will allow GFP being washing out from the beads. Now we let the GFP get into the collection tube number 3, which means that we wait until the entire volume passes through. When everything is done, cap the column and observe the solutions inside all 3 collection tubes and the column under the UV light. Of course, you will need to use the protection equipment, the face shield. Now you can see that GFP is accumulated in the collection tube number 3, and there is practically no GFP in the collection tubes number 1 and 2. Now you could make your conclusions based on this photograph.